Perfect. Good morning, everyone. My name is Horacio Lopez-Nicora. I'm a new assistant professor in the Department of Plant Pathology. I joined the department last year in August. Before that, I have been working in South America, in Paraguay and Colombia. So if you do have trouble maybe understanding something due to my very strong Southern accent, just raise your hand virtually and I'll make sure to rephrase and make sure that we are all on board with what I'm going to present today. So what I want to do today is stay in the rhizosphere, in the area that surround the soybean root, and we're going to talk about the Soybean Cyst Nematode, arguably the most important soybean pathogen in North America. And then we're going to transition to talk about other soil-borne pathogens that affect soybean roots and by doing so, damaging our crop. So let's go ahead and start with what I have planned for today. We're going to talk first about the Soybean Cyst Nematode and understand a little bit of the biology of these pests to understand how we are losing yield. We're going to talk a little bit about the management or the integrative management that we propose to mitigate the problems that this pathogen cause. We're going to talk a little bit about what's the status of Soybean Cyst Nematode in Ohio and learn a little bit about our project, Know Your Numbers, funded by Ohio Soybean Council, United Soybean Board with the SEN Coalition. And we're going to talk some of the, options, alternatives that we're bringing to growers to take advantage of. We're going to talk about soil sampling and processing samples for Soybean Cyst Nematode in our lab. Then we're going to transition and talk about other soil-borne pathogens, some true fungi, but also some water molds, Phytophthora and Pythium mainly. And with each of these organisms, we're going to touch a little bit on seed treatments as an alternative for managing them. So with that, let's jump into the fascinating life cycle of the Soybean Cyst Nematode. Heterodera glycines is the scientific name. It will overwinter as eggs in the soil. These eggs can be loose or it can be enclosed in the dead body of the female that we call cyst. Now, the numbers of eggs per 100 cc of soil is the data that you want when you're sampling for Soybean Cyst Nematode. Is the numbers of eggs, not the numbers of cysts, what will give you an indication of the initial inoculum in that field. When conditions are favorable, these eggs, and you can see that some of the nematodes are wrapped inside those eggs, they will hatch into this second stage juvenile. This is the only infective stage of the Soybean Cyst Nematode. With the aid of this stylet on the head of the nematode, it will be able to penetrate the soybean roots. So here we're staining the root and we're seeing how the nematodes get inside the root and they will migrate, causing physical damage already to the root. They will migrate to the vascular cylinder. In the vascular cylinder, the nematode will start probing the cell, trying to engage the soybean in an interaction. If the soybean is susceptible, those cells around the head of the nematode will start feeding the Soybean Cyst Nematode. At this point, the nematode will lose motility, will become sedentary, and will not be able to move anymore, and will start feeding and enlarging in that same spot. So now the root have transformed those cells, becoming into feeding sites and feeding the nematode so it can complete its life cycle. The male will regain this worm-like structure and will get out of the root to fertilize the female. And now the female will be able to lay, on average, 250 eggs every 24 to 30 days in a growing season. So the female will lay eggs in the soil that now can hatch again and begin the cycle, and this is during the growing season. But eventually, the female will start filling up with eggs and dying. Once the female dies, her body is filled up with eggs, and that dead body will become the cyst that will protect the eggs to overwinter. So clearly, the nematode is in the root modifying these cells, making the plant believe that in those cells are the progeny that the plant has to feed. The plant is putting all the energy now, feeding the nematode that is on the root. And when stressful conditions take place in the field, we can see symptoms like short plant, stunted plant. It will occur on patchy areas. It will be aggregated in space because the distribution of the nematode is also aggregated, clustered in space. When we have other type of stress, such as nutrient deficiency, for example, let's say potassium deficiency, we will see that the symptoms that those nutrient deficiency will cause, let's say marginal chlorosis, will be enhanced in the presence of the nematode. Now here's a problem with this, because we're going to see symptoms that resemble potassium deficiency. We will suspect, as good agronomists, that we have nutrient deficiency. We're going to take a soil sample, send it to the soil lab, and the results will come that we actually do have potassium deficiency. We will make the adequate corrections, and we will alleviate this stress. However, we did not send part of that sample to a nematology lab. So by correcting that nutrient deficiency, the plant will stop showing that type of symptoms, but the nematode in the field will continue increasing its population until the numbers are extremely high that we will definitely see symptoms such as short plant, very low yielding areas. And when we actually consider checking for soybean cyst nematode, those numbers sometimes are extremely high. That is hard to get them down. So take-home message here is that we do not always see visible symptoms with soybean cyst nematode, and therefore we should be always proactive in identifying if we have the problem. If we plant a susceptible cultivar next to a resistant one, we can see those type of symptoms. However, if the field is heavily infested and we only plant one variety, which is what we normally do, we will not be able to detect that difference because we don't have something to compare with. So yes, we do have symptoms. Most of the time, they happen when there are severe or very stressful conditions already in the field, or when the population is extremely high, that later on it will be hard to get them down. So we can also see the pathogen. We can go and pull some roots, tap the soil, look at the root, and we can see these white lemon-shaped, pearlish in color females on the root. Very small, but still visible to the naked eye, not to be confused with the nodule that I'm pointing here. But this is the best way to diagnose that we actually do have the problem. So there's a lot of research out there from different trials across the Midwest region that soybean cyst nematode can reduce yield between 15 to 50 or more %. And really, a lot of the research that we're doing and we're showing over and over is that this reduction can actually take place with absolutely no symptoms, which for me is a nightmare. So here what I'm trying to show in these images is that, yes, sometimes if we compare a resistant line to a resistant line to a susceptible line, we can see some symptoms and we can quantify that yield reduction. However, in many cases, and I'm going to show some data for this. But Terry Niblack, soybean nematologist, retired from Ohio State University in 2003, started showing that if you put a susceptible variety next to a resistant line, and start measuring everything to try and actually quantify a symptom like plant height, branching, intensity of the foliage, there was absolutely no difference between resistant line and susceptible line. But when you went and harvest the susceptible and the resistant, you could actually quantify that huge yield reduction. And again, the importance of how adapted nematode populations are in the Midwest, that they can reduce yield with absolutely no visible symptoms. So what do? We have a huge problem here that can cause up to or more 50% yield reduction, and we don't see it. We think that we're growing the best soybean in town, and what should we do? Well, we should know if we have the problem, and we're going to talk a little bit about that with our Know Your Numbers project. But once we do have the soybean cyst nematode in our field, we will not get rid of it. Eradicating soybean cyst nematode from a field is nearly impossible. So we have to manage the pathogen. In 2005, Niblack suggested these 3 R's proposal of managing: rotate, rotate, and rotate. So the first rotate goes for rotating crops. This is the soybean cyst nematode. It cannot complete its life cycle. It cannot reproduce on a non-host that is not soybean or other host. So in this case, corn is a perfect example of what we can use to rotate and get those numbers low and mitigate the problem. So the soybean cyst nematode will not cause any problem in corn, small grains, and sunflowers, and other non-hosts. So crop rotation is always a good management and the beginning of a good and long-term management program. You have high numbers of SEN, rotate to a non-host crop. However, we know that if prices continue to being so good for soybean, and if really that's what we want to do, then we should consider the other 2 rotate. So rotate resistant soybean varieties. So even though we have in more than 90% of our SEN resistant variety commercially available, this source of resistance, PI 88788, we still should consider rotating varieties, planting different type of genetic background in our field, not get the nematode used to the same food over and over again. So rotate resistant soybean variety, and then try to rotate the source of SEN resistant. And as I mentioned just now, more than 90% of our soybean cyst nematode resistant variety that we have out there commercially available have this source of resistance, PI 88788. We're going to talk a little bit about that. However, there are others that we can find in some varieties. So also rotate and try to choose cultivars with other sources of resistance, such as Peking, for example. Okay, but the best way to manage soybean cyst nematode, and this still does not change, is by means of resistance. If we have soybean cyst nematode, and we do have soybean cyst nematode as our main limiting constraint, we should consider planting a soybean variety. It doesn't matter what source of resistance, just put a soybean variety there that will help start that long-term process. Now, here comes something that we have been observing more and more, and we should be aware of, because we are strongly recommending that resistance should be the way to do this. But we know that soybean cyst nematode, the populations that we have in this part of the US, continues to adapt and reproduce on PI 88788, that source of resistance that is very prevalent in most of our commercially available soybean cultivars. So what do I mean when I say continues to adapt and reproduce? So soybean cyst nematode was discovered in the United States in the 50s, 1954. Soon after, many places that grow soybeans started detecting the presence of the pathogen and reduction in yield. Soon after, of course, soybean breeders starting screening and identifying different sources of resistance. One of them, PI 88788, very easily introgressed and deployed in high yielding soybean varieties. So at the beginning, this was great, because for the population of soybean cyst nematode that we have, that wild type AG type 0 or race 3, with cultivars that had PI 88788, we were doing a very good job at managing this problem. The nematode could not reproduce, or if it did reproduce, it reproduced below 10% compared to a susceptible soybean. However, you can imagine for over 30 years, using the same source of resistance just exerted this selection pressure on the population of the nematode, shifting its virulence. So now our population, AG type 2 or race 1, is able to reproduce on PI 88788 above that 10% compared to a susceptible. And now from different surveys across the different states in the north central region, we know that more than 85% of the soybean cyst nematode population actually can reproduce to some level on that PI 88788. So this is a problem that we're seeing. Some populations can reproduce more than others, so we can still benefit from this resistant source. However, understanding this, we need to revisit this rotate, especially rotating these sources of resistance and incorporated other non-hosts that can be economically beneficial for us as well. We also need to protect our seed in some manner by using nematodes protectants. Again, this will not be the solution, but will be something that we will add to our resistant program. And then maybe incorporating cover crops. This is something that I'm very interested in researching, and my master's student, Ali Pierce, is actually starting to look into the different cover crops that we can grow in Ohio and see how the status on host viability to soybean cyst nematode is. So which one can allow reproduction of soybean cyst nematode, which cover crops can really not allow the reproduction. Alternatively, understanding the mechanisms that cover crops have will help us use these to mitigate the SEN damage. For example, some cover crops will attract the nematode and allow the nematode to get inside, so it will serve as traps, but the nematode will not reproduce. So this will be a great information to have when we select what cover crops to put in our field. You know, I'm putting here a chart that I found from a great nematologist, Greg Tilka from Iowa State, and Caitlin Bisonet at that time. Many of the products that probably you are familiarized with that can be used for managing other problems, but also can help to protect our cultivars to soybean cyst nematode. So Ilevo is one of those, fluopyram, that we're going to talk a little bit when we cover SDS. Okay, so what I want to do now is I want to show some data that we have, just to, you know, show the importance of using resistant in a heavily infested field. So what we did in this project in 2015, this was a 3-year experiment, actually, 13, 14, and 15. We have resistant soybean lines and susceptible soybean lines. These are BC4. These are almost near isogenic lines, very homogeneous genetic background, only that the resistant had the source of resistance for soybean cyst nematode, that PI 88788 source of resistance, and the susceptible lines did not. We purposely put these lines in a field with a population that can actually reproduce on PI 88788. Again, the main question here was, can I still benefit from using a resistant line with PI 88 source of resistance in an AG type 2 population field? So on the x-axis, I'm showing the amount of soybean cyst nematode at planting in X per 100 cc of soil. This is log transformed. That's why the numbers here are very, very low. This is just for the analysis. And then on the y-axis here, I'm showing the soybean yield in bushels per acre. Two important things that I'm sure you're realizing here. And this will happen in every field. As my number of soybean cyst nematode increase in the field, my yield will decrease. And that happened both for the susceptible and the resistant line. The other important thing that I'm sure you're noticing is that there's always this gap between resistant and susceptible. And you can see as we are increasing soybean cyst nematode, that gap is becoming greater. So if we just get the average of resistant and susceptible, and we just plug in this simple exercise here, we realize that by choosing a resistant line in a field that had soybean cyst nematode, we were increasing our yield by 8 bushels per acre. At the soybean price at that time, 2015, if you recall, we are essentially losing $77 per acre if we decided not to plant a resistant variety in that field. With the price of soybean today, that's what, $110 per acre? Pretty significant. And now here comes another part of this project. We had 800 plots for this research. We went and we took every single measurement we could to try and actually evaluate if we could see any symptoms between resistant and susceptible line. And we didn't. We measure height, intensity of the foliage, color, branching, nothing, absolutely no symptoms. However, we detect yield reduction. So similar trial that or type of study that we did in 2021. This is in Waterman Farm. We went and in a field that have high levels of soybean cyst nematode, we put a grid. These are 30 by 35 foot grid. And we put a flag in the center of each grid where we went and we took a composite sample to quantify X per 100 cc of soil at planting, because that number will relate very well with yield reduction. The blue quadrants are ones where we did not detect soybean cyst nematode. 0 X per 100 cc of soil, we call those non-detected. And then the very red one was the one that we actually detect soybean cyst nematode in high numbers. So above 1,000, 3,000, 5,000, and we call those high SCN. At the end of the season, we went and we got yield in bushels per acre. And again, we did several measurements during the season, and we absolutely see no symptoms in this field. So what I'm doing here to show you the data is I'm using a box plot. I like this type of graph because the wider the box, the more variable your data is. The tighter the box, the less variability you have in that data set. Here on the x-axis, I have those two classifications. So non-detected, those blue quadrants where there was absolutely no nematode detected at planting, and then the high levels where everything that had above 1,000 X per 100 cc were used. So I put two lines here, and this indicates the average for those plots. So you see when there is no soybean cyst nematode detected, that those plots were yielding 57 bushels per acre. However, in field above damage threshold, we see that on average, those fields were yielding 35 bushels per acre. Again, if we plug in that very simple exercise, we can see that we are losing 37% of yield in very high infested fields with SCN. And that range falls between that 15 to 50% that we talked at the beginning of the presentation. If we're pretty generous with price, you know, we can see that when we have soybean cyst nematode above damage threshold, you know, we can get to $280 per acre of yield reduction, and that's huge in my opinion. Okay, so you know, we keep doing this type of experiment just to validate how damaging the soybean cyst nematode is. And I want to share with you what is the status of the soybean cyst nematode in Ohio. With funding from Ohio Soybean Council and the United Soybean Board through the SCN Coalition, we have been conducting a survey, receiving soil samples from growers all over Ohio from 2018. And the data I'm going to show is the summary up to last year. But we're continuing this survey, and we're going to talk more about this and how you should take advantage of this survey. So we receive samples and we process them for SCN testing, and we count how many X per 100 cc of soil we can detect. So if any sample had any soybean cyst nematode at all, we will classify that as positive, but then we want to actually know the number of soybean cyst nematode. We need to know the numbers because those levels will help us decide on a management strategy. So more than 60% of the samples that we received, almost 750 samples processed so far, had soybean cyst nematode. Now, you know, such a negative news, you know, I want to always follow by a positive result, and that is that of those samples that had soybean cyst nematode, fortunately, most of them had very low levels of SCN. Low levels that we can easily keep it down or even bring it to levels that will not cause damage to the nematode, to the plant. Sorry. Remember, it's easier to maintain low levels of SCN than to bring these very high levels down. So we do have fields, unfortunately, in Ohio that have soybean cyst nematode way above damaging threshold. And I know you're asking yourself, Well, of those nematode, have you evaluated what's the virulence profile of those nematode? Can we use, that PI 88788 that is present in more than 90% of our commercially available soybean cultivars out there? And yes, and we continue doing this in the greenhouse. So far we did 60 of those very high levels of SEM. We put it in the greenhouse. It takes 1 month for us to evaluate the virulence profile. And what we see is that 10% of that is the SEN type 0, the wild type, which means that for those 10% of those samples, we can use any cultivar that says resistant to soybean cyst nematode, and we will do a good job at managing it. However, more than 85% of those samples that we put in the greenhouse can develop on PI 88788 above that 10% compared to a susceptible. Okay, so above 10%, is 11% something that we should be worried about? No. And you should continue using resistant to managing that problem, but consider maybe rotating and using a different source of resistance. The problem is that some of this population can reproduce on PI 88788 60% compared to a susceptible. So now we have only 40% efficiency, if you think it that way, on managing soybean cyst nematode. So we should be aware of this, and this is an information that as a grower, is very valuable when we need to make a decision in what cultivar we're gonna use. Okay, so I'm talking about this survey funded by Ohio Soybean Council and as part of the SEN Coalition funded by United Soybean Board. This year, we will continue doing this test. We will continue receiving soil samples from growers, and we will process them for SEN. So if you're wondering if you have soybean cyst nematode in your field, there are two important ways that you can answer that question. Go dig up a root and see if you actually find the nematode. The other one, which I consider easier, is to go and collect a soil sample. And you will do this exactly the same way that you will collect a soil sample to send it to a soil test lab. So we recommend, breaking fields in areas no larger than 20 acres. Do that zigzag pattern collecting subsamples to make a composite sample that you will then mail to the nematology lab. We recommend choosing areas that are very uniform or alike. If you're going to sample a low area, make sure that that composite sample comes from a low area, and then take another one maybe from a high region of that field. Sometimes when we suspect that we have the problem, we recommend that a more strategic sampling should be done. Where does soybean cyst nematode increase more in number? In areas where we have high pH, nematodes will reproduce more. So maybe target those areas for a soil sample. We know that the only way that nematode will get into your field is by us introducing it through maybe contaminated, infested equipment. So maybe those field entryways will be a place to sample. We are receiving up to 2 samples per growers, and we're processing these free of charge. So, when spring comes and you can take a soil sample, or during the season, if you suspect that you have soybean cyst nematode, or, after this presentation showing that, there's absolutely no symptoms and the inoculum is just building up for later to drastically reduce your yield. And if you're curious, take a soil sample and send it to a lab. We recommend sometimes to take a soil sample from a low yielding area and another one from a high yielding area and submit those samples and then compare the numbers of soybean cyst nematode that you receive. But, you know, up to you. These two samples are for you to use the best way that you want. If you have two fields, take soil sample from those fields, send it to the lab, and we'll process those. The results will come to your email that you will provide with the numbers of soybean cyst nematode per 100 cc of soil, a level that we will assign, either moderate, high, low, or trace, and then a table below that with management recommendation. And of course, by contacting your extension educator of your county, or you know, giving me a call or an email with any questions, that's also a best way to reach out and learn more about this. So I think this is a really good opportunity, and I hope you take advantage of this this coming season. Okay, I want to stay around the root of the soybean, but I want to transition now to other pathogens that can threat our production. I always say, you know, it's not that we have, that's my field with only soybean cyst nematode, and over there I have my field with Fusarium, and back there is my Phytophthora field. It's always a mixture, and it will really depend on how we transition through the season and the environmental conditions for one of these, you know, to take over and actually cause a lot of problems. So we need to be just aware that we can encounter several pathogens in the same field. So we're driving, passing through our nice soybean field, and we see these areas again aggregated, this patchiness of leaves wrinkling up, drying prematurely, and then we get closer and closer to that area, and we see these very characteristic symptoms where the leaf is dying, but the veins remain green. This is sudden death syndrome, SDS, caused by Fusarium virguliforme. Actually, it's caused by several Fusarium species, SDS. However, in the United States, Fusarium virguliforme is the most prevalent one. It is a root pathogen. It will be in the soil. The fungus will overwinter in the soil. It will grow with no problem in the soil. It will overwinter also in soybean debris that was infected the previous season. And now more recently, with new evidence of research from other universities, we know that this fungus can infect corn without causing problems in corn. However, increasing that initial population of Fusarium in the field. It will infect the soybean seedling right as soon as the seed germinate. Conditions that will require is cool soil temperatures below 65 Fahrenheit, wet conditions, and compaction will enhance the severity of this problem. It is a root pathogen, so most of the damage will occur and take place in the root system. So if we dug those roots out, we will see that some of those roots that are required for water and nutrient absorption will be dark and dying or dead. Sometimes under conducive environmental conditions, we will actually see the fungus, this bluish color, you know, mycelia and spores growing on top of the surface is the fungus itself. We will pull our knife and cut that taproot, and we will actually see that is not clear, white, and healthy. The root is this color, in this case, an indication that we have this problem. During the season, the fungus will continue damaging the root and by doing so, reducing yield, but will also produce toxins that will be translocated to the above ground portion of the plant. And those toxins will produce that very characteristic symptoms. The fungus is not here. If we go and collect these samples and send it to a lab, we will not be able to isolate the fungus from those symptomatic leaves. If we spray anything to this leaf, unless that product reaches the root, we will not be able to control the pathogen. These symptoms are due to the toxins that the fungus is producing on the root of the plant. Again, like with other pathogens, under the presence of soybean cyst nematode, the severity of SDS in the field will also be enhanced. So most of the time where we do see a lot of SDS, that may be a good indication that soybean cyst nematode can also be present there. A good way or strategic way of soil sampling to see if we have the nematode there, just, enhancing the problem. Those leaves, of course, as you can imagine, will defoliate prematurely, helping that yield reduction that is taking or that it's already taking place with the root rot. So how do we manage SDS? It will be an integrated management. We know that there is quantitative resistance to this fungus. So we see a plot here, highly susceptible cultivars compared to the plot behind where there's a high level of resistance. And we can see that that's something that we can use in our benefit. Managing soybean cyst nematode will help. There is evidence out there that there is an interaction between these two organisms where both becomes more severe when they both are co-infesting the fields. Drainage will help reducing compaction. Crop rotation is a good management, however, not with corn. And it's funny, this part of the presentation, because, for the past 40 minutes, I've been telling you, if you have soybean cyst nematode, plant corn. And now here I'm saying, yeah, maybe do not use corn. And the reason is that we're learning that this fungus can increase its population in corn without causing any problems to corn. But then when soybean comes after, we have built an inoculum for that. So here I'm showing a picture where, half of this field was under a soybean corn rotation in heavily infested field with Fusarium virguliforme, the SDS pathogen. And then on this other half, a different type of rotation scheme was used by using oats and other small grains instead of corn. And you can see that there's more symptom here in this portion. Seed treatment is a great way to manage SDS. However, we need to choose the effective active ingredient. And there's evidence that fluopyram is one that is working very well. And that's illegal. There's other ones such as Saltrol that seems to have really good management for SDS. And we are going to, do some of these seed treatment trials this summer. So I'm very excited to start working with these and maybe next year come and present on that research. But again, there are other organisms out there, right? And we just need to know what we have and what we need to deal with. So some other pathogens that are or can cause problems to our seedling are different species of Fusarium. So one of them is Fusarium graminearum. And probably if you hear Pierce Paul presentation on wheat, you will hear a lot about Fusarium graminearum affecting wheat headscab, right? And then if you hear Pierce Paul talking about problems in corn, you will also hear that Fusarium graminearum can cause ear damage and reduce yield. And now here I'm talking that that same Fusarium graminearum can also cause problems to our seedlings in the field. Rhizoctonia is another one. Sometimes require a little bit of a warmer spring than we're used to, so sometimes we don't see much, but if it's out there and conditions are favorable, it will definitely cause problems in the very early stages. So some data that Anne shared with me is this one where they're testing fungicides for true fungi, and we're going to talk a little bit about what does that mean. But essentially grabbing seedlings and inoculating with Fusarium graminearum, and then go and see that percentage of seedling affected. When there's nothing, no treatment at all, we see that seedling affected is very high compared when we actually use a product that can control a true fungus. So in this case, I'm showing some of the QOI, the Strobis. So we have a control where there's no fungus infecting the seedling. Then you can see how the seedlings just grow perfectly well. Of course, there's no problem there. And then I have here some where we actually put the pathogen and we infest these seedlings, but also we treat them with a fungicide for true fungi, in this case, Captan and Maxim. And we can see how that actually takes really good care of our seedlings, where we do not see any difference compared to the control. However, when we put the pathogen, we expose those seedlings to Fusarium graminearum, and we use a product, right? Apron. You're probably familiarized with apron, used to control water moles. So this product will not take care of the true fungi. We can actually see how Fusarium graminearum, even though these seedlings have been treated with a quote-unquote fungicide, it's not being effective, right? So we really need to know what active ingredient we're using for what specific organisms. And I think this picture lets very well us to our next talk here, which is the water moles, right? So the two most important water moles that we will encounter is Phytophthora and Pythium. They will cause this reduction in stand, pre and post emergence dumping off. If some of the plants will survive this threat, this early threat, but are not resistant later on, when we have conducive environmental conditions, plants can completely collapse due to this pathogen. I'm showing the life cycles very similar for both of these. They require seeding water. Water moles produces a zoospores which swims in water to reach to the root and cause the problem. Compaction will also be a problem. Recall that compaction was also a problem for SDS, wet conditions. But this is what I want us to focus. If we have Phytophthora in that field, Phytophthora will do very well at 60 degrees at infecting and causing problems. We will probably also have different species of Pythium that do very well at low temperatures, very cool spring. They will infect and cause problems either by killing the seeds or the plant just as it emerged. Other species of Pythium do very well at warmer temperature. So I'm showing here a situation where no matter what the condition is for that spring, we will encounter a problem if we have, saturated soils, heavy soils, and a history of these type of problems. Another very cool research that was done in Anne Doran's lab and published in 2020, same soil from a field, but incubated at different temperatures. The same soil incubated at different temperatures. You will retrieve different type of species of Pythium and Phytophthora. So if it's a cooler temperature, that will benefit certain species, and when you have warmer temperature, others will, be more prevalent. So in other words, we have no choice but to deal with an array of different water moles that we need to take care of. Moreover, some of these isolates are able to reproduce on our sources of resistance. So that leads into a transition of, the need to use seed treatment, but the need to use the adequate seed treatment for the adequate pathogen. So we see here these three active ingredients that are meant to control water moles that still work very well, excellent for the water moles. However, they're not recommended if you're trying to manage true fungi. And the example of apron, if you remember that picture with Fusarium graminearum, just shows that. On the other hand, fungicides meant to control true fungi are excellent at doing so, but not recommended if water mole is the problem. Of course, again, it comes to the same idea that we have multiple pathogens threatening our soybean production, and therefore a combination of different active ingredients in our seed treatment will actually protect our soybean better. With that, and because I'm almost out of time here, I want to just finish this presentation by making some final considerations. Know the history of your field, and of course we do, right? We know what pathogens we have, we know what we struggle with. Now with the options of sending your soil sample for Soybean Cyst Nematode, you will actually be able to know that as well. But know the history of the problems you have, and that will make you decide on what seed treatment to use. Play with soil temperature, but if you hear most of Laura Lindsay's results, we don't want to go and wait a lot to plant, right? So that's a fine line to play. So maybe to be safe, use a good and adequate seed treatment. Do we practice reduction in tillage, or we do a non-till? Maybe that will call for a seed treatment. Do I have a history of Phytophthora and other type of true fungi? Maybe we want to do a combinations of active ingredient seed treatments. And then evaluate the seed protectant that we will use with Soybean Cyst Nematode resistant source if we do have that problem in our field. And with that, of course, I want to thank the people that help a lot in my lab: Ohio Soybean Council, United Soybean Board, and USDA NIFA. And I'm here for any questions. Thank you.
Thanks, Horacio. We do have a couple of questions. One was pertaining to your nematode presentation. Can I pay to have soil samples tested at the OSU lab if I have more than 2 samples?
Yes, definitely. Take advantage of these two samples, right? Free of charge. And then, yes, we do provide that service. It's $20 for each of that sample tested, and you will get your numbers of Soybean Cyst Nematode back in an email, like I explained, with recommended management. I also say that we do in the greenhouse that virulence profile. That does take more time. Sometimes we need to build inoculum from the Soybean Cyst Nematode from your field to get to the level that we actually will need for screening. So that normally takes longer time, but we also offer that service if anybody wants. We are doing this as part of the survey, so we hope to get more of this information out there next year. So we have another question.
Yes, Dick asked, if soybeans are followed with wheat, then you put out double crop beans, do you get the same increase of cyst as if it had just been planted to be beans 2 years in a row without wheat in between?
Okay, yes, this is a great question. It's something that actually I'm very interested in evaluating, and I will make it part of my program. So we do have a lot of information about crop sequencing from other states. In Illinois, for example, we did a lot of work where we just plant corn. And sometimes I have conversations here when I'm hearing, well, if I just plant corn continuously, I will do it until I get rid of soybean cyst nematode. Unfortunately, that is not the case. We show that first year where you rotate to a non-host, you do see a very large reduction in soybean cyst nematode. But then later, it seems that it's just stabilized and just, you do not get the same reduction. So you always want to include soybean as part of that rotation to stimulate that egg hatching. So a resistant soybean variety is very important here. Now, your question specifically was the double crop. And that's something that Southern Illinois have been doing, and it's something that I want to evaluate here. So we know that the nematodes will hatch at a correct temperature, like when the temperature gets warm. But we also know that some of these eggs will hatch when they sense root exudates. So one of the ideas here is to actually, have the eggs exposed to different root exudates that will continue hatching, and by so reducing the numbers for next season. So for Ohio, I don't have this data, but next year I will be able to answer that with our data from Ohio.